Welcome, John Carroll University students. You are taking General Biochemistry 431. It's been a really interesting week, traumatic in some ways. Some of you have left University Heights and went home. This has just been kind of unexpected. We didn't know this to the beginning of the week, and then it just became a snowball effect, and now we have moved to online courses indefinitely. And I think we can do this. I think we're going to make this a good experience, and bear with me. Let me know any concerns you have via email. I think that's probably the easiest way. There is a Canvas chat, but an email seems to be the quickest way to send information. We can later look at Canvas chat or other tools that are available in Canvas, and we will try to make use of that. I'm in the learning process as well, and I think we're going to be able to do this. So jumping right into where we left off Monday, we were into metabolism and energy transfer from small molecules such as ATP. We also kind of blend this in with small cofactors such as NADH and NADPH. And so maintenance reactions for ATP, we've talked about this. I'm just reviewing this to get you up to speed. For example, creatine phosphokinase, NDP kinase, adenylate kinase, should know those obviously for the test coming up. These are maintenance reactions that maintain high levels of ATP and or energy. All enzymes that do this. Let's go ahead and take a look at some of that. Some of these enzymes. Creatine phosphokinase catalyzes a reaction of creatine with ATP. Phosphoryl groups of ATP are transferred to the nitrogen of creatine. We're going to see this time and time again where nitrogen will accept different molecules, different small molecules such as carbons and phosphoryl groups, oftentimes on a phosphate. Creatine phosphokinase, highly expressed in muscle cells, not so like a liver cell, but in a muscle cell, it's going to be highly expressed. It allows creatine phosphate to serve as a very rapid source of energy, a storage mechanism of providing high energy molecules and energy to the different metabolic reactions needed in the cell. Allows ATP formation to remain effectively constant despite rapid change in energy demands. It's a near equilibrium reaction at NER. It is therefore not a reaction that's highly regulated except by the substrates and products. Here we see the reaction itself. We see the nitrogen that will act as the nucleophile. We see the mechanism down here where the phosphate, the phosphoryl group is attacked at the phosphate, being an electrophile. And then we see the breaking of the bond here on the ADP molecule, which is formed here. And then we see phosphorylated creatine formed here, which stores part of the energy from ATP and can immediately affect enzymes and donate a phosphate group as well and keep the energy levels high or provide energies that are necessary in the cell for metabolic purposes. Energy coupling with ATP. NDP kinase, this is a somewhat indiscriminate enzyme. We haven't seen that. You've been told in the past that enzymes are very specific for their substrate. Now you're being told that, wait a second, this NDP kinase can actually phosphorylate ADP, CDP, UDP, GDP, and TDP. All nucleotides, very important in the cell. It's what makes our RNA, our DNA. It also, these ATPs, CTPs, well, ATP not so much, but CTP, UTP, GTP, TTP, TDP, etc. are often signal molecules. Okay, irreversible reaction, and as we've seen in the previous slide, we have to note whether it's reversible or not. The previous reaction we saw was reversible. In this case, NDP kinase, irreversible for good reasons, because of the compartmentalization of these different nucleotides that do different things in the metabolic pathway. It's probably good from a perspective of regulating different metabolic pathways that it cannot easily go back and forth with NDP kinase. Adenylate kinase. Now we're looking at a near NER reaction, near equilibrium. This reaction here can go back and forth. It is metabolically, it is reversible as opposed to the NDP kinase reaction we saw. Near equilibrium in the NER reaction provides a means of rephosphorylating a low energy AMP. AMP is also a signal molecule. You can imagine it can form cyclic AMP, which is another signaling molecule. We've seen that previously with G proteins, and the release of calcium by a signal on the extracellular surface of a cell results in 2 ADP products. So we get some useful energy out of ATP and AMP coming together in this linear fashion, and the enzyme adenylate kinase does this. It lines up the substrates just right so the reaction can take place much more easily. Without an enzyme, you wouldn't have something there to hold the substrates and make them easily align up for a possible reaction as we have here. We have inhibitors. Ironically, you can read this. It talks about diadenosine tetraphosphate, and it talks about these inhibitors, diadenosine pentaphosphate, and it basically is another way to inhibit enzymes. This particular enzyme. We're giving you a specific example. This inhibitor obviously is effective for what? The enzyme that we're talking about. And there is, laboratory support to show that this can occur, that you can get inhibitory responses from using different designed inhibitors. For example, this has a 5-phosphate. The substrate itself only typically has 4 phosphates involved in the entire reaction. And so let's move on here. And NADH, electron carriers can be energy intermediates between catabolism and anabolism. Principal electron carrier is an NADH molecule. Changes in redox states involve 2 connected processes, always 2 reactions. We can look at them as half reactions, and oxidation is a loss of 1 or more electrons from a molecule. Reduction is a gain of 1 or more electrons from a molecule. The 2 processes must occur together. There must be something that is oxidized, something that is reduced. Alcohol dehydrogenase is an example of an oxidoreductase enzyme where oxidation and reduction must occur together and simultaneously. Showing the half reaction here, ethanol going to acetaldehyde, forming an aldehyde from the alcohol. 2 electrons and 2 hydrogens are given up. The effective equivalent of 2 hydride, a hydride ion, which would be a hydrogen with a lone pair of electrons and a negative, or an electron and a negative value. The NAD plus is reduced and gains a hydride. In reality, the electrons are transferred with a hydrogen, or as a hydrogen ion, a hydride ion, excuse me. The mechanism for alcohol dehydrogenase, we see NAD plus here coming in after the enzyme using zinc cofactor. We haven't seen that yet. Zinc being positioned in the enzyme to help stabilize the substrate, interact with the enzyme and the substrate simultaneously. This is all happening in the active site. The substrate, for example, is being manipulated so that it can basically, a hydrogen can be removed, a hydride, if you will, removed to NAD plus, which becomes NADH. We then have the hydrogen removed, and we'll have these electrons come over here, and we'll form the aldehyde group, okay? So you're going to see these mechanisms throughout. You produce a reduced NADH molecule from NAD+, and so hopefully you should be able to understand this mechanism. NADH, not the only electron carrier. NADPH is a hydride carrier as well. Has an extra phosphoryl group, that is the only difference. And it's attached to the ribose in the nicotinamide adenine dinucleotide molecule on the sugar that's involved in this molecule. And it is, you'll see in the pentose phosphate pathway, the sugar pathway, NADPH is generated and useful in that metabolic pathway. Most enzymes use one or the other, be it NADH or NADPH, for the oxidation reduction reactions as one method for hydride transfer or electron transfer, if you will. So it's metabolically irreversible what this transhydrogenase enzyme can do. And so it can provide utility, allowing NADH and NADP plus to share. When NADH is high, it is able to add to NADP plus and make that a reduced molecule to do further work. You notice we don't see NADPH. We don't see the reverse. This is an irreversible reaction where NADH is donating its hydrogen, its redox state to NADP plus, becoming NADPH. Maybe the cell for some reason has some demand for NADPH, and that's why this is occurring. So if you can think of that, it is an irreversible reaction. It's probably not a near equilibrium reaction. This is a good thing to look over. Go ahead and do so. As you know, NADH is produced in glycolysis and the Krebs cycle, and used in electron transport chain, as we'll find out later, and as you've seen in some of your other biology courses, and helps to produce ATP via oxidative phosphorylation. Plant chemistry also involves a lot of NADPH. NADH and similar nucleotides, everything here pretty much is the same for NADH and NADPH. NADPH has this, at the 2-hydroxide, a phosphoryl group. So you see how where the hydride here, this is the reduced form, this is the reduced form, this is the oxidized form, this is the oxidized form. And the aromaticity gets reestablished when it loses a hydride. And we see positive charges on the oxidized form. So if you see a positive cofactor that's NAD plus or NADP plus, you know that it is missing a hydride. You should be able to understand these structures. Not too complicated. If I ask you a question on the test, you should be able to sort of understand what's going on here. Mobile cofactors and the pathway view. These cofactors are a link between and or within metabolic sequences. When we think about parallel pathways, perpendicular pathways, meaning they cross between different metabolic pathways. Connections between the reactions often are viewed from a pathway point, and a pathway involves a product of one reaction becoming the substrate of the next reaction. Stuff you already know. It's the main point here, simplified. These cofactors are mobile, and they are intricate and important and integral in many metabolic pathways, not just one pathway. They are pretty much kind of ubiquitous in many pathways, and we'll see that throughout. Here's an example of a role of a mobile cofactor helping this metabolic pathway, whatever it might be. Would it be possibly glycolysis in the energy investment phase? Could be. And then we can see a pathway that could actually run perpendicular through this pathway and run off into a tangent. Maybe we have a a need for a small molecule somewhere else in the body in a different metabolic pathway, and so that can be siphoned off from this main pathway into another pathway crossing through it, if you can imagine that. Here we have mobile cofactors in a pathway view. The HMP shunt slash the pentose phosphate pathway. And glycolysis. These are two pathways that are where? They're in the cytosol. They are not in the mitochondria, right? Okay, so we need to know where we are when we're talking about which certain metabolic pathway. Where are we in the cell? Sometimes that's a really easy question to answer. Sometimes maybe not so. One thing, side note here, they're talking about prosthetic groups. Those are, for example, we've already seen that with hemoglobin, which is a large net-like molecule that can hold oxygen. We'll see that in other enzymes as well, especially in pentose phosphate pathway and glycolysis, perhaps. Just overall, thinking about a pathway, the first substrate in a pathway can be the final product of a pathway, previous pathway. So the first substrate can actually be a product, obviously, from another metabolic pathway. I don't like how this is worded. I'm going to change this. So listen to what I say rather than reading this is important, but it's redundant. I need to fix this. If the pathway is linear, the sequence, then there is one rate usually for the entire pathway. Why? There will be a rate limiting step. You've heard about rate limiting steps in general chemistry. That still applies to biochemistry. Although more complicated in how the enzyme is ran, the rate limiting step will determine the pathway flux speed. And that will also be a regulation point. The slowest point often, of course, you would think would be the regulation point, and it is. So these mobile cofactors go throughout different metabolic processes, be it glycolysis and pentose phosphate pathway. So they can go back and forth in the cytosol from glycolysis to the pentose phosphate pathway. Okay, so what do we have here? Yes, we are having a pathway here from B1 to B4. It's shut down. If it's shut down, it cannot use the NADH, assuming it shuts down at least by B2. NAD plus cannot be formed. NAD plus, which is a oxidizing reagent, helps to oxidize A2. It becomes more reduced NADH, the cofactor, making A3 a more oxidized molecule. So when you shut this down, you can effectively shut down pathway B and then eventually slow down or shut down pathway A. That has biological consequences, biochemical consequences, and that's the gist of this slide. The pentose phosphate pathway takes place in the cytosol of the cell, the same location as glycolysis. The two most important products from this process are the ribose 5-phosphate sugar used to make DNA and RNA, and production of the NADPH molecule, which helps with building other molecules in anabolism, anabolic processes.